Hi, Dr. Biology here. This is a exam paper commentary for the AS Biology Paper 1 from 2016. Paper 2 has the same structure as Paper 1, so there is one comprehension question and there are short answer questions for the rest of them, and again it's 1.5 hours, and they'll test you for all 4 topics. Okay, on to the first question, and it says about endo and exopeptidases and how they're involved in the hydrolysis of proteins. That's related to the spec number 3.1.4.2, related to proteins and enzymes. And the question says, name the other type of enzyme required for the complete hydrolysis of proteins to amino acids. The answer is dipeptidase. They would accept membrane-bound dipeptidase as well. Question 1.2, it says suggest and explain why the combined actions of endo and exopeptidases are more efficient than exopeptidases on their own. It's worth 2 marks. So they'll want you to talk about either one of those, but endopeptidases hydrolyze internal bonds, whereas exopeptidases, they remove amino acids or hydrolyze them at the ends. So therefore it would be more effective because there'd be more ends, or it would increase the surface area to allow hydrolysis to occur. Here is the answer. There we go. Notice they say they'll accept break for hydrolyze. Well, I would try to use the word hydrolyze. That's the more correct terminology, and that's really the terminology you're going to need to use in the full A level. Question 1.3, and it shows the co-transport mechanism for the absorption of amino acids into the blood by a cell lining in the ileum. So straightforward co-transport, you can see sodium and amino acids. Okay, probably you might have looked at sodium and glucose, but it's exactly the same. It says the addition of a respiratory inhibitor stops the absorption of amino acids. It says explain why. Well, first of all, respiratory inhibitor, you need to know that that means no ATP is going to be produced, therefore you can't release energy. So if there's no release in energy, there's no active transport. So, explain why. Well, you've got 3 marks to explain why, so you've got to use the information that they give you. Okay, well the first thing we said is that no ATP is produced. Okay, so no ATP. So what will happen? Well, if you look at the diagram, what will happen is that sodium ions are no longer moved out. So sodium is not moved out of the cell. Alright, so that's the second point. That's because they need to be actively transported. Now, the question though talks about the absorption of amino acids. So if sodium's not moved out, then there will be no concentration gradient for the sodium to move into the cell with the amino acid. Okay. You can mention that there's no active transport as well, because that wasn't in the stem of the question. So if it's not in the stem, you can mention it. So that's fine. Sodium ions not moved out, so there's no diffusion gradient for sodium to move into the cell with the amino acid. Okay. Again, you can see that they'll accept these things. If you were just to say no active transport of sodium ions, that's 2 marks, because again, if you look back at the question, they want to know how all of that will affect the absorption of amino acids. So it's important you read the question carefully. Question 2, and it talks about the distribution of stomata on leaves. Okay, so it talks about 2 species, and they removed small pieces from the lower surface of the leaves and they mounted them on a microscope slide, then counted the number of stomata in several parts of the epidermis using an optical microscope. And it says, suggest appropriate units the student should use to compare the distribution. Okay, well, that's going to be apparatus and techniques. Okay, so you would have used an optical microscope, but also you might have looked at gas exchange as well and looked at structure of stomata. So you can have a variety of answers, but you're looking at millimetres squared, so you're looking at the surface area. So it could be stomata per millimetre squared or centimetre squared, and they'll accept other permutations as well, as you can see. It wouldn't be per micrometer squared, okay, it'd be per millimetre squared. There we go, and also amount of stomata is incorrect. It's because you're comparing the distribution of stomata, so it's going to be in a view on the microscope. In terms of looking in the optical microscope, it's going to be millimetre squared. This next question is about using an optical light microscope. So this is within spec 3.2.1.3 about principles of optical microscopes. It says the piece was very thin. Explain why this is important. So in the stem of the question, it says that it's using an optical microscope. Okay, now an optical microscope, the specimen's light has to pass through. Okay, and also it's thin, so there's only a single layer of cells. Okay, so you don't want too many overlapping. So there we go. So that would be the answer. Notice they wouldn't give you the mark if you said single layer without reference to the cells or tissue. Okay, so you couldn't just say there is a single layer. You'd have to say there is a single layer of cells or tissues. So just be careful on that point. Question 2.3, it says give 2 reasons, okay, why it was important that the student counted the number of stomata in several parts of each plant tissue. Well, it's quite simple, because if you look at the distribution of stomata, I mean, you might have a greater distribution in one area of the leaf than another area of the leaf. Okay, so in that case, what you need to do by doing several parts is you have a representative sample, okay, and the reason being is that you are trying to obtain a reliable mean. Question 2.4 asks about the fact that the plant was a xerophyte. Okay, it says suggest 2 xerophytic features of the leaves that the plant might have other than distribution of stomata. This is in 3.3.2, where it specifically mentions in the spec, it says structural and functional compromises between the opposing needs for efficient gas exchange and the limitation of water loss shown by terrestrial insects and xerophytic plants. So there are lots of different features. Okay, so you could have many different answers here. The key ones are, I think, if you've looked at things like xerophytic plants such as marram grass, you'll know that stomata are in pits, or that cacti, for example, they have a thick waxy layer, thick cuticle. Some of them have rolled up, folded leaves, so they trap water vapor, or spines, so they reduce the surface area, or last one, hairs, so they trap water vapor. Now for the 2 marks, you've got to mention, okay, both. I mean, if you look for 1 mark, you need to say what the structure is and what it does. Okay, all right. It's important that you put both points and you get 1 mark for each of those. So notice it says so, and so forth. Okay, so it's really important you follow that when it says suggest and explain. So suggest is the first thing, explain is the second, and you get 1 mark for each suggestion and explanation. And then question 2.5, it says that they then compared the rate of transpiration from the two species. She did this by measuring the rate of water uptake by each plant species, and then it says suggest 2 reasons why the rate of water uptake by a plant might not be the same as the rate of transpiration. So this question is testing your knowledge about what is water being used for in the plant. Okay, so this, it's not just being used, it's not just being released by transpiration. So there's quite a few answers you could have. Just because it just needs you to suggest, you just need to say what water is being used for in the plant. So let's have a look at the answers. Okay, so it can be used for support or turgidity, it can be used in photosynthesis, used in hydrolysis, or water is produced during respiration. So in terms of the spec, it's kind of asking where water is being used in a plant, and you would have covered that in several different areas. Question 3, it says a tubifex worms are small thin animals that live in water. They have no specialized gas exchange or circulatory system. Okay, so they're invertebrates, don't have lungs, etc. Probably have spiracles. Now, probably you don't know what a tubifex worm is, but that's fine. They've described what it is, and it actually shows a picture of it. It says, question for 3.1, name the process by which oxygen reaches the cells. Well, very straightforward. Oxygen can reach cells through diffusion. Okay, it's a straightforward diffusion. If you said facilitated diffusion, that would be wrong because it doesn't use protein channels. Just a reminder, this question is related to gas exchange. Okay, so this is going to be gas exchange in the tracheal system of an insect or similar invertebrates. So this is 3.2.3 in the spec. Right, question 3.2, it says use the information provided to explain how 2 features of the body of the tubifex worm allow efficient gas exchange. Well, let's have a look at the question again. It says they are small and thin animals. Okay, live in water, that's fine, but I think small and thin, if we're going to talk about gas exchange, those are the 2 key things. All right, small and thin. So how does that allow gas exchange to occur? So remember, with this type of question, you can't just say thin or thin membrane. You've got to say, well, why? So thin membrane, so there is a short diffusion pathway because that allows efficient gas exchange. Or you could say it was then thin, so it has a large surface area to volume ratio. So if you look at the diagram, actually, you can see that it has a large surface area to volume ratio. So those would be the answers. Okay, then they give you some information in 3.3 about tubifex worms and where they live. Okay, and there's a few clues in terms of they live at the bottom of ponds. Okay, and it says the partial oxygen is low. It says pollution of water by sewage can cause the partial pressure of oxygen to fall to below 0.2 kPa. Figure 3 shows the oxyhemoglobin dissociation curve, okay, for tubifex, as you can see. Now, this is going to be related to mass transport, so this is actually spec number, it's 3.3.4.1. Okay, so there's some important information down here that says basically tubifex worm has 50% saturation with oxygen at 0.08 kPa. Question says explain how this, in other words this, enables this species to survive in water polluted with sewage. because notice they did talk about partial pressures of 0.2 kPa in the stem of the question. Okay, so as you can see, there is a high or 50% saturation with oxygen below 0.2 kPa. So 0.2 kPa is how it's is the saturation with sewage, whereas the Tubiflex can operate between 0.08 and 0.2 kPa. And why? Why is that important? Well, oxygen is needed for respiration. Okay, so that will allow it to survive. 3.4 says the species lives in ponds, lakes and rivers, cannot survive in seawater. And it says use your knowledge of water potential to explain why they cannot survive in seawater. Okay, well, if it's related to movement of water or transport of water, we're talking about water potential, then we're talking about osmosis. So in terms of the spec number, this is in 3.2.3. What I find interesting about this question is that the question can ask you, the bits of the questions can ask you different parts of the spec. So when you start a question, don't expect all the questions to be related to that part of the spec. Okay, you've got to look for the clues to what it's about. So this big clue is water potential. So the idea is related to water potential. So if it was in seawater, okay, the seawater would be more concentrated. Therefore, the water potential is higher in the worm. You can't say high, all right? Or you could say there's a lower water potential in the seawater. And then you've got to explain that why it cannot survive in seawater is that the water would leave, and you can say this, by osmosis, and you have to say it as well. Now, it's interesting in the stem of the question, they mention water potential but not osmosis. So obviously you can mention that in your answer. So it's quite a nice question, that, okay? Just talking about relative water potentials. Notice you can't say, as I said, you've got to say it's higher or lower water potential. You can't say high or low water potential, it won't be correct. Question 4, so they give you lots of information here, and they talk about the effect of different types of animal farming on the diversity, clue to what part of the spec it is, and the number of good old dung beetles. I do like a good dung beetle. They determine the number of dung beetle species and their total, sorry, I'll say that again, the number of dung beetles. Oh no, sorry, didn't read it properly. The number of dung beetle species and their total number on intensive I, rough grazing R, and O organic farms. So they got some results. So you got the mean number of species in this one, okay, and the type of farm. Notice, okay, they've done some statistical tests here, so that might come into the question in a minute. And then the mean total number of dung beetles, okay, and the type of farm. So they've looked at standard deviation, okay. Now this is related to biodiversity within a community, so talking about species richness, number of different species in a community. In this case, this community is related to agriculture or different types of agriculture. So the spec number is 3.4.6. So 4.1, what is the mean species richness for dung beetles on rough grazing? So we need to look at this one, okay? Alright, so species richness, you should know, is a measure of the number of different species in a community. On rough grazing, if we read the graph through, and you'll get 14. It's as simple as that. Then goes on to say, in addition to the information, what other measurement is required to calculate an index of diversity? Okay, so if you know your calculation for index of diversity, you would need to know the number in each species, okay? So number in each species. Question 4.3 then goes on to talk about the standard deviations, and it says, explain what the standard deviations suggest about the differences in the mean total number of dung beetles between the different types of farms. 2 marks. Okay, so let's have a look at the data. Okay, so difference, not that one, that one. Right, so if you look on the mean total number of dung beetles, alright, you look at the standard deviation. So if I just show you standard deviation here, okay, that one, that one, that one, you can see there is no overlaps at all. Okay, so they are significantly different. Alright, so if there's no overlap in standard deviations, then it is significant. What's interesting about the answer to this question is that it doesn't want you to describe, okay, so be very careful to read the command word, which is explain. So it wants you to talk about the standard deviations. It doesn't want you to give any data, okay, it doesn't want you to use the graph to describe how the number of dung beetles in each of the different types of farms. Question 4.4, it says the scientists placed traps to collect the dung beetles at sites chosen at random. And it says explain the importance of the sites being chosen at random. Well, very simply, there is no bias. That's all it's looking for. Okay, so no bias in the sampling. That's all you need to put. Okay, question 4.5, it says, on the intensive farms, the farmers had removed the hedges to increase land for grazing. This resulted in a decrease in the diversity of birds. It says, explain why the removal of hedges causes a decrease in diversity of birds. And it's 3 marks, so it's related to biodiversity. Now, you need to realise that if you remove hedges, increase land for grazing, you're going to remove the number of species. Okay, so if you remove the number of species of plant, there'll be less food or fewer food sources, and particularly there might be less places for the birds to have nests or shelter, so therefore there'll be fewer habitats. This answer is a pretty good one in terms of anything related to biodiversity. So I know they're talking about farmland and hedges, but you could be talking about much larger ecosystems. Okay, so removal of tropical rainforests, for example. All right, so it's always the removal of species due to fewer food sources and fewer habitats or niches. That will come up as well in your Year 13 work when you do a large section on ecology. Okay, question 5, and there is a lot of information again in this question, so you need to read it carefully. So it tells you that seeds of some plant species require chilling before the embryos they contain can grow, so they've got to chill them before they'll grow. During chilling, storage molecules in the seed that contain phosphate are broken down, and phosphates are then transported to the embryo. Scientists investigated the change in mass of phosphate in the embryos of cherry seeds at 2 different temperatures, and you can see the mean mass of phosphate in the embryo and at 5 degrees and at 25 degrees, and I can't quite see it, but you might be able to, the number of days. No, the number of weeks, of course. Okay, so you can see there's quite a big difference between 5 and 25. So 5.1, it says phospholipids are one of the storage molecules, and those are broken down to release phosphate. Well, in terms of biological molecules, regardless of the biological molecule, when you start to break things down, that is known as hydrolysis. Okay, so that's related to biological molecules. It's a biological molecule, and it will be hydrolysis. 5.2, it says the scientists concluded that an increase in phosphate in the embryo was linked to the growth of the embryo. And it says suggest 2 reasons why an increase in phosphate can be linked to the growth of the embryo. Okay, so how can phosphate increase the growth of the embryo? Okay, so you've got to put your thinking hat on here, really. You've got to think, well, where are phosphates? Okay, so where are phosphates in terms of growth? Well, the first one is the structure of DNA and RNA, so you're going to need those kinds of things for growth to occur. That is the spec in 3.1.5, okay, and you need to know that both DNA and RNA contain a phosphate group. So another place that phosphates are important is in ATP. So ATP contains an inorganic phosphate group, and obviously that is required for growth because it releases energy. We also know a phospholipid contains a phosphate as well, so that's going to make cell membranes. And I've just noticed the last marking point for phosphorylation. But to be honest, if you are doing AS, you may not have discussed that yet, because that's really a year 13 section on respiration. So you may have known that through extra reading around the subject. Also, there's additional marking points. You might want to mention phosphates in NADP, AMP, and RuBP. But again, that's related to other aspects, respiration, photosynthesis, in the year 13 work. So the main ones would be RNA, DNA, ATP, and also makes membranes. You can't say phosphates make phospholipids. You've got to mention what it makes, and it's making the membranes. Question 5.3, it says calculate the ratio of the mean mass of phosphate found at 5 and the mass of phosphate found at 25 at 9 weeks. Okay, so let's look at the graph. I've copied the graph over. So we've got to find 9 weeks, which is kind of, it's difficult to do on this machine, but it's about there. Okay, all right, so we can find the mean mass of phosphate by drawing a line across. Okay, there, and very, very roughly, I'm afraid, there. So I'm going to go for the easiest. I'm going to say that's 1 and that's 4. Okay, so very simply, if it's asking for a ratio, the ratio I'm going to put is, well, actually it's slightly above 4, so I'm going to say 4.1 to 1. Okay, so I would go with the 1 because it just makes your life easier. So you can have a range of 3.7 to 1 to 4.1 to 1. Okay, so it must be shown as a ratio like that. Luckily, okay, it's quite easy to do just by reading the information off the graph. You only get 1 mark, but it's not that hard to do. Question 5.4, and it says the chilling requirement of seeds is considered to be an adaptation, okay, for survival, with seasonal changes in the environmental conditions. Suggest how this adaptation, okay, may enable the species to survive and respond to seasonal change. It's 3 marks, okay. So in a natural system, okay, you want the seeds or embryos to remain dormant, okay, so you don't want them to be active during the winter, okay, so it allows them to survive the winter or cold. Also, you could mention that the embryo, the embryo starts to grow during the winter, ready for the spring, okay, and it develops during the spring. And then it will obviously emerge during the warmer times when there'll be greater photosynthesis, more light, for example, and then eventually it would produce more seeds and offspring during the growing season to allow it to survive. It's important that you know that plants do not hibernate, okay, so don't use that word hibernate, okay. But yeah, the seeds are remaining dormant, or they're starting to grow and develop, as they have done the cherry seed, for example, and they then will develop at the correct time of year, which is in the spring. I would say that there's more light during the spring, therefore more photosynthesis, and therefore seeds will germinate. Okay, and it then produces more seeds that allow it to survive it through the growing season. Question number 6, and it talks about FEV1, which is forced expiration volume. The volume of air a person can breathe out in 1 second. Then it talks about emphysema as a lung disease, and then it says scientists investigated the effect of aging and long-term cigarette smoking on FEV1 and the development of emphysema. Figure 7 shows the results, so you can see FEV1, percentage mean value, okay, and how it increases, and you can see on the bottom that is the age in years. Okay, so you can see difference between non-smoker and a long-term smoker, and you can see that where the symptoms of emphysema start and the severe disability. So you can immediately see the younger you are, the higher your FEV1. Okay, but as you increase in age, okay, your FEV1 decreases, but it decreases at a faster rate in long-term smokers than it does non-smokers. So spec-wise, this is 3.3.2 and about ventilation and the exchange of gases in the lungs. Okay, now you may not have been taught about emphysema, but it tells you all the information in the stem of the question. Question 6.1, it says that they determined the mean FEV1 for 25-year-olds, and it says suggest 2 precautions that should have been taken to ensure that this mean value was reliable. Okay, so this is related to scientific methodology. Okay, so how did they make it reliable? So you could have many different things. One thing they didn't talk about was sample size, so you need a large enough sample size. You also need to choose individuals randomly. Okay, you need to check that they are healthy in the beginning, so we're looking at the effects of smoking and non-smoking, so we want to know that they're healthy otherwise. Possibly an equal number of males and females, and definitely you want to repeat the readings. Notice in the notes you could mention how many people, so if you say 20 plus, that's a large sample size, and you could say sex or gender rather than equal number of males or females. Question 6.2, it says explain the importance of determining a mean FEV1 value of 25-year-olds in this investigation. So if you're finding a mean, you're trying to see if there is a comparison. Okay, so you're finding the mean. Is there a comparison between the 2 groups? And what are you looking at? You're looking at the effect of smoking particularly, or you could say age or emphysema, or it takes into account as well if you've got any anomalous results. So there's 2 key things there that come up time and again, things about reliability and also the importance of doing a mean to do a comparison. Okay, so those kinds of things, they'll come up in different types of questions, but those I've seen many questions over my time that look at those kinds of things, so you can relate that to other topics. Question 6.3, it says the mean value of non-smokers decreases after the age of 30. Okay, so it's not related to smoking, and it says it wants you to use your knowledge of ventilation to suggest why. So when you look at the mechanisms of ventilation, you should know that the internal intercostal muscles are less effective with age. Or you could say that the lung tissue, there's less elasticity or less elastin, or there's less recoil, okay, related to lack of elasticity. Then question 6.4, it says one of the severe disabilities that results from emphysema is that walking upstairs becomes difficult. And it says explain how a low FEV1 could cause this disability. Okay, so obviously if you're finding it difficult to walk upstairs, there must be something going on in terms of oxygen uptake and respiration within the muscles. So if you have a low FEV1, it's not just less oxygen, you've got less carbon dioxide being removed. Okay, so that increases in the blood. Therefore there's less oxygen or uptake of oxygen, so therefore less respiration. Okay, or you could say there's more anaerobic respiration. They will also accept other answers about more lactic acid as well building up, which would make it more difficult. Okay, question 7, and it says that HIV attaches to a specific protein receptor on a helper T cell. A low percentage of people have a mutation of the CCR5 gene. It's something that you wouldn't have been told in your teaching and being taught. And that codes for a protein receptor. This mutation results in a non-functional protein receptor. It says explain how this mutation can result in the production of a non-functional protein. And you get 4 marks for doing that. Okay, so this is related to mutations, gene mutations, which involve a change in a base sequence of chromosomes. In your spec, that will be in 3.4.3. Okay, and it wants you to talk about mutations in the production of this non-functional protein receptor. So, as I said, it's a change. A mutation is a change in the DNA base. Okay, so you could talk about deletion or substitution or addition if you wished, as it doesn't say that in the stem of the question. What result does that do? Well, it changes the amino acid. Therefore, it alters the hydrogen or ionic or disulfide bonds, which then changes the tertiary structure, making it non-functional. Okay. Again, it's quite a common answer, that. Okay. The key difficulty there is that when the amino acid is changed, it does alter the bonds, and you need to mention some of those bonds, because those changes in those bonds will change the tertiary structure, which is important to state, of the protein. So 7.2 says people with the CCR5 mutation show a greater resistance to developing AIDS, and they want you to explain why. Okay, so we need to think about the receptor. All right, so when there's no mutation, the receptor site is complementary to the virus. Okay, so the HIV virus, whereas here it obviously is not complementary, so it can't bind or attach. Alright, so if it can't bind or attach, as we know with AIDS, basically, HIV actually, there's no replication of the virus and therefore no destruction of T cells. So this is related to your knowledge of cell immunity, okay, and also knowledge of HIV, but it's mainly about cell immunity and the fact that receptor sites are going to be different, therefore no replication. Question 7.3, it says the frequency of the CCR5 mutation is highest in Europe. Scientists have collected data on the history and number of HIV infections in Europe. Using these data, scientists have concluded that the high frequency of the CCR5 mutation is not due to natural selection. Suggest 2 reasons why they reached this conclusion. Well, this is related to genetic diversity and adaptation, which is 3.4.4. For this question, you need to know something about HIV and AIDS. Okay, so you need to know that HIV has only been present for a short time period. So in terms of natural selection, your knowledge of natural selection, you'll know that it takes many hundreds, if not thousands, if not millions of years. Okay, so it's not been present. It's only been present for a short period. And the CCR5 gene has probably been around for many years. It could be advantageous as well, but also you could argue as well maybe there's lower exposure to HIV as well in Europe. So you can have those answers, okay? Okay, this next question, 8, is related to one of your required practicals. So required practical 6, okay, where it was looking at aseptic techniques to investigate the effect of antimicrobial substances on microbial growth. Okay, so it tells you that they looked at 4 different concentrations of the same disinfectant, grew a culture on the agar in a petri dish, and then they cut out 4 filter paper discs, soaked each disc in one of the 4 concentrations, and then placed them in the nutrient agar, 25 degrees, 24 hours. And then there are the results. So, first question, and it says, explain why there is a clear zone around each paper disc. So for the mark, you've got to say that the bacteria are killed or destroyed. You can't say there was no growth in that area. It's looking at that there was basically the bacteria are killed. So the next question is about the scientific methodology, and it says that they researched information on this disinfectant prior to carrying out his investigation. So the student used a maximum concentration of 40%. It says use the figure 8 to explain why. Okay, so let's look at figure 8. Okay, so if you look at the clear zones, the last thing is like in D, for example. If, for example, he used a higher concentration, there would be a larger kill rate of bacteria. So therefore D would spread into A, okay, and A might be larger, and then C, there'd be overlap. So basically the clear zone would be too large, or they would overlap, or you could argue if you used a massively big concentration, there would be no bacteria on the plate. So you wouldn't be able to compare the different treatments. So there are the answers. So it's the idea that the clear zones would be too large. .3 goes on to talk about 2 variables the student should control. Okay, so control variables in the filter paper discs in this investigation. So the filter paper discs need to be the same size. Okay, so you could accept thickness, area, diameter. They've got to be made of the same material or absorbency, and also be in the solution for the same time period. Okay, notice the things that they won't accept. So you can't say same shape, as the shape is shown on figure 8, and you can see that they are the same shape. Also the reference to the volume of disinfectant as well. Okay. Okay, 8.4, it says use the areas of the clear zones in figure 8, which I've got here. Okay, so I've just copied it down. Alright, you can see that there. And it says determine how many times more effective concentration D is than concentration B. So you're going to need to do pi r squared on each of them. Now, since I don't have the paper in front of me, I'm going to kind of make it up as I go along, but you would measure it, okay, and then that's the diameter. Okay, so diameter, whatever the diameter is, you divide it by 2, Okay, so for example D, I'm going to say the diameter was 40, so the radius is 20. I'm going to do that 20 squared times pi, so pi r squared, and that will give me 400. And then C, okay, I'm going to imagine the diameter is 24, so the radius is 12, and you square it times pi, and that will give you 144. Okay, and then it says how many times more effective, so all you do is 400 divided by 144, and you will get the answer 2.7 times. So there we go. So you'd actually use the paper, work out the pi r squared, and divide D by C, and that will tell you how many times more effective it is. Question 9. Table 1 shows the volume of blood in a woman's left ventricle at different times during 1 second. Okay, so it's related to the heart. Okay, gross structure of the heart and understanding pressure, volume, volume of blood, for example. So that is mass transport in animals. Spec number is 3.3.4.1. Okay, question 9.1, it says use table 1 to calculate the heart rate in beats per minute. Okay, so we'll go from 0, and you can see the volume of blood, so we need to find 1 beat. So if we go down the data, okay, so let's go down the data until we get a repeat. We get a repeat at that point, so that's at 0.9, so that's 1 beat. But we need to know beats per minute, so there are 60 seconds in a minute divided by 0.9, and that will give you 66.7. So you're rounding up 66.6 recurring, so it's 66.7. Question 9.2, it says the stroke volume is the volume of blood pumped out of the left ventricle during 1 cardiac cycle. And it asks you to use table 1, which I've copied down here, to determine the stroke volume. So determine the stroke volume. So you take the maximum amount of blood per centimeters cubed and take it away from the minimum. Alright, so that will give you a stroke volume of 70 centimeters cubed. Question 9.3, it says some people produce a much higher ventricular blood pressure than normal, so high blood pressure. So this causes tissue fluid to build up outside the blood capillaries, and it says explain why. So the clue is in the question. If there's a higher blood pressure, then more fluid is going to be forced out of the tissue fluid, okay, and less will be returned due to the high pressure. It's important that you talk about the fluid, okay, because it's tissue fluid. It's you don't want to talk about plasma being forced out, it's the fluid. You don't want to say more tissue fluid is forced out either. You just want to say that more fluid is forced. They will accept water for fluid as well. So several answers that you could have there. Question 9.4. Some drugs are used to reduce high ventricular blood pressure. Some drugs used to reduce high ventricular blood pressure cause widening of blood vessels, so dilation of the blood vessels. Suggest how the widening of the blood vessels reduces ventricular blood pressure, and that's 2 marks. So you're going to use your knowledge of blood vessels and know that if you have a larger lumen, okay, so the larger lumen, the space in the blood vessel, this reduces the pressure. You could also say there's less friction or resistance, but the top 2 ones would probably be the ones that you would go for in the exam. Okay, on to the last question. Now this question is split into two and it's worth 10 marks. 10 marks is pretty much a grade boundary, so it's quite important that you really give these a good go. This first one is really nice. Describe. Okay, if you've done your revision, if you've actually gone through the spec and you've made sure you understood everything in the spec, you're just literally saying, okay, how the structures of starch and cellulose are related to their functions. So this is in 3.1.2. It's all about carbohydrates. These are structural carbohydrates, and it wants you to talk about their functions. Because it's split into starch and cellulose, it's important to state that you'll get 3 marks for talking about starch and 3 marks for cellulose. There's no need to contrast, you're not contrasting, okay, but you do need to give reasons for the shapes, the structures. Okay, so for example, starch is helical. Why is it helical? So it is compact. That will get you 1 mark. Okay, so as you can see, starch, large, insoluble, so osmotically inactive. Okay, you could say it doesn't affect the water potential. The branched, so glucose is released for respiration. It's large, so it cannot leave the cell surface membrane, so any of those 3. And then cellulose is long and straight, made of beta glucose. They're joined by hydrogen bonding to make them surprised actually I haven't got the same with cellulose in terms of the reason for them. So joined by hydrogen bonding basically to keep them again compact, form fibrils, which provides rigidity and strength. So those are the possible answers. All of those things would be in your textbook, would be in your notes, and in your revision. It's important you revise all these kinds of details. Question 10.2 says describe, yet again for 5 marks, the processes involved in the transport of sugars in plant stems. So that is related to mass transport in plants. That's 3.3.4.2. Okay, and it's about the mass flow hypothesis for translocation in plants. Okay, so the kinds of things you need to say is sucrose is actively transported into the phloem by companion cells. This lowers the water potential in the phloem and water enters by osmosis. Notice you need to mention that water enters by osmosis. There's a higher pressure, therefore mass flow, or transport towards the sink or roots. And at the sink roots, sugars are removed. Okay, so notice it's about plant stems as well, the movement in plant stems. There are many other things you can get in there as well. Okay, so again, if you've learnt about mass flow transport in plants of sugars, then you should be okay. So here are the total grade boundaries, so for both paper 1 and paper 2, so it's out of 150 marks, and you can see that the grade boundaries are pretty low. For an A you only needed 59%. You do have to put this into context though. This was the very first linear new spec paper in 2016. I think many students at that time found it difficult, and also I know teachers were getting to grips with the new spec as well. But you can see that, you know, going through, I noticed going through the mark scheme, they're looking for really specific answers. Okay, so in terms of suggest and explain, so if you're going to say something, you need to explain it as well. You also need to know your calculations, and you need to know your stats tests. But there were things in that paper, particularly paper 2, there's a lot of description, and in paper 1, so you know, in terms of content you could have gained quite a few marks from it. Anyway, so that's that really. So I hope you found that useful. I am shortly going to go through the AS paper 1 and 2 of 2017, so please do subscribe for more Doctor Biology exam commentaries. Thanks for watching this video, and I will see you soon.